Today, we're diving into something pretty amazing: how scientists are using supercomputers to hunt for cancer that's hiding in plain sight. It's a story about designing a virtual key to unlock one of the disease's biggest secrets. So, let's just start with a huge question. How do doctors find cancer they can't see? I mean, this isn't just a riddle. It's one of the single biggest challenges in medicine today, and it's the exact problem that this incredible new research is trying to solve.
And the focus here is on prostate cancer. It can be notoriously tricky to track, especially when it starts to spread, metastasize to other parts of the body. These tiny groups of cancer cells can be too small for normal scans to pick up, basically leaving doctors flying blind. And that uncertainty, well, it makes treatment incredibly difficult. So, the mission, when you boil it down, is pretty straightforward. Make the cancer cells light up. The idea is to create a special molecule called a radiotracer that you can inject into a patient. This molecule is designed to hunt down and tag only the cancer cells, making them glow on something like a PET scan. It's like giving doctors a map to the enemy. Okay, but to create a tag that specific, you need a unique target to aim for. You need something on the cancer cells that isn't really on the healthy cells. And luckily, scientists found one. They discovered a specific protein on the surface of prostate cancer cells that acts like a very specific lock. That target is called prostate-specific membrane antigen, or PSMA for short. Now, the most important word here is overexpressed. It's not that healthy cells don't have any PSMA. They do. It's just that prostate cancer cells have a ton of it, sometimes hundreds of times more. And this really shows you what we mean. Look at that. A healthy cell might have a few of these PSMA locks just hanging out on its surface. But a prostate cancer cell? Whoa. It is absolutely covered in them. And that huge difference is what makes PSMA the perfect, unmistakable target. So you can see where this is going, right? If we can design a molecule, a key, that's shaped perfectly to fit into that PSMA lock, then we've got our homing device. We just attach a tiny radioactive signal to that key, and it will swim through the body, ignore the healthy cells, and bind right onto the cancer, making that invisible enemy suddenly very, very visible. Now, here's where the story takes a really futuristic turn. Finding a molecular key like this would normally take years of tedious work in a lab, testing thousands of different compounds. But this time, the entire search was done in silico, which is just a fancy way of saying it was all done on a computer. That's right. Using some incredibly powerful software, researchers didn't just search for existing molecules. They designed brand new ones from the ground up. They started with a known chemical structure called Glu-ureido, and then began tweaking it, changing it, and optimizing it virtually to create the most perfect key possible for that PSMA lock. And this virtual process was super systematic. First, they designed 60 totally unique keys. Then, they ran binding tests. The computer actually simulated how tightly each of those keys would fit into the PSMA lock. It also checked for safety, analyzing how toxic each molecule might be. And finally, it ranked all 60 of them to see which ones came out on top. So, they started with 60 brand new digitally crafted molecules, each one a potential breakthrough. Now, imagine trying to physically create and test all 60 of those in the lab. That would be a massive undertaking, costing a ton of time and money. But that's the beauty of this approach. After all of that intense virtual testing, checking how well they'd bind, how stable they'd be, how safe they'd be, the computer narrowed that big pool of 60 all the way down to just 6. The 6 most promising candidates of the bunch. So yeah, the virtual search worked. It was a huge success. By using all that computing power, the researchers could skip a whole lot of the slow, expensive trial and error and just focus on the ideas that had the best shot. So let's meet these virtual winners. And here they are, the top 6. You can see the key finding for every single one of them was an excellent binding score. What does that mean? Well, in our lock and key story, it means the computer predicts these keys will fit the PSMA lock perfectly. They'll slide right in and, just as important, they'll stick around long enough for a PET scanner to get a really clear picture. And check this out. This is a quote from the conclusion of the research paper. I want you to notice 2 words here: diagnostic and therapeutic. That means these keys might not only be able to carry a signal to find the cancer, but they could also potentially carry a tiny payload to treat the cancer. That's a true 2-for-1 punch.
Okay, so the computer has done its job. It took this massive, complex problem and handed us a very short, very promising list of solutions. But what happens next? How do you get these molecules off the screen and into the real world? Well, this is where the work moves from the digital world to the physical one. The next step is for scientists to actually synthesize these 6 molecules in a real lab and start testing them, first on cancer cells in a dish, then eventually in animal models. It's still a long road, but this in silico head start makes that journey so much more direct.
And this is the big picture, why this matters so much. This whole approach could completely change how we discover new medicines. It makes the process way faster. It makes it cheaper. It helps scientists focus their energy on the ideas most likely to work, which could accelerate the path to getting new cancer treatments to the patients who desperately need them.
So we'll leave you with this final thought to chew on. We always think of big scientific breakthroughs happening in a lab with microscopes and beakers. But as this research shows, maybe, just maybe, the next great leap forward in the fight against cancer won't start in a lab at all, but on a laptop. Thanks for joining us.